Hello students, welcome back to VA NEET Academy. In our previous classes we have seen in detail about endoplasmic reticulum. So today's class is about Golgi apparatus. Now imagine a factory which has produced its product. Now what has to be done with the product? The product has to be packed, labeled and then delivered to its particular designations, isn't it? So this is the function which is being carried out by our next organelle called as Golgi body. So Golgi body is also called as the packaging and dispatch center of the cell. So in today's class, let's see in detail about Golgi bodies, its function, structure, and how does it help in producing and labeling and packaging the materials to different locations. It does not produce, but then it receives the produced product. So the product which is produced by endoplasmic reticulum, that is the product called as proteins and lipids, will be received by this Golgi apparatus and it will be transported to its designated destination by Golgi apparatus. So let's see in detail how does it do that. Thank you. Hello students, welcome back to VA NEET Academy and let's get started with our class. So today's class is about Golgi, the right pronunciation is Golgi. Okay, some pronounce it as Golgi, some say it as Golgi. Actually it is Golgi apparatus, the packaging and dispatch center of the cell. So we will look in detail about the Golgi apparatus in today's class. Okay, so let's see the definition first. Definition of Golgi apparatus. Okay, now this is a membrane bound cell organelle. Okay, this is a membrane bound cell organelle. Let me fix it so that it's clear for you. Yeah, this is a membrane bound cell organelle that is responsible for modifying, packaging and secreting. Okay, modifying, packaging and secreting the proteins and lipids. Now this is the definition of Golgi apparatus. So this is the organelle where the modification, packaging and secretion of the protein which is formed earlier in endoplasmic reticulum and lipids are being processed. Okay, so this is about the definition of Golgi apparatus. Now let's come into the discovery of Golgi apparatus. Now this Golgi apparatus was discovered by a scientist called as Camillo Golgi. Okay, now this is also called as Golgi body, Golgi complex or else Golgi apparatus. Okay, either way it all means the same. Clear students? Okay, so let us move on to the structure of Golgi apparatus. Structure of Golgi apparatus. Okay, so this consists of, actually when we talk about the structure, I need to tell you it is made up of two basic structure called as cisternae, okay, and vesicles. Cisternae and vesicles. So what is a cisternae? Cisternae is nothing but you can say it is a stacked pile of
plates. So it looks like a stacked pile of plates. It looks like a stacked pile of plates. That is called cisternae. Okay. So vesicles, they are nothing but a small sacs. Small sacs which carries the modified and packed proteins. Clear, students? Okay. So this Golgi apparatus has two phases in their diagram. One is called the cis phase and the other is called the trans phase. Okay. Cis phase is also called as the receiving end. Okay. And the trans phase, you can also call it as a shipping end. Right? Okay. Now what is a cis phase? Cis phase is something which faces the endoplasmic reticulum. Okay, this faces the endoplasmic reticulum. Clear? And receives the protein and lipids from the endoplasmic reticulum, and it gets entered into the Golgi apparatus. So you can also easily remember this as an entry gate. So this can be an easy analogy for you to remember. Cis phase is nothing but an entry gate. So from where the vesicles carry the proteins and lipids from the endoplasmic reticulum, and this is why it faces the endoplasmic reticulum side and comes into the Golgi apparatus. Trans phase, these sends the processed material to their final destination. Final destination. So you can say trans phase is from where the processed material gets into the final destination. You can easily remember this as an exit gate. The easy analogy for this will be an exit gate. So trans phase is an exit gate, cis phase is an entry gate. So let us now try and draw the diagram, a very simple diagram. It has got only few things to remember. Okay, supposingly this is the stacked pile of plates. What are they called? I told you what are they called, the stacked pile of plates. Remember? So you can call this as a cisternae. Okay, apart from cisternae, it also has the vesicles formed here and there, which gets secreted out from the cisternae. Okay, so for example, now this is one cisternae from where the vesicle is forming. It's getting formed, and this is the place from where the vesicle is getting fused inside the cisternae. Clear? Okay, now let us try and label this diagram. As all of you know, this is the cis phase and this is the trans phase. So cis phase is near where? The endoplasmic reticulum. Okay, now this is the incoming vesicle because it is coming inside and getting fused into the cisternae. You can see it is getting fused here. Okay, and this is the outgoing vesicle. You can see here, from here the vesicle gets popped out and gets out of the Golgi apparatus and is taken into the final destination. Clear? And these are your cisternae, the pile of plate kind of structure. Okay, the inside of the cisternae is called as lumen. The tubule, the inside of the tube is called the lumen. So this is the simple basic diagram of a Golgi apparatus. Okay, now let us talk about the functions of Golgi apparatus. functions of Golgi apparatus. Okay. So the first function is obviously a very important function, modification of proteins. Okay. So the proteins which are being received from the endoplasmic reticulum is modified in this Golgi apparatus. After modification is done, it is the other function which it carries out is packaging of proteins and lipids. Everything which is received by the Golgi apparatus is being packed also. Packed in the form of what? It is packed in the form of vesicles, or else it is packed inside the vesicles, as I have shown in the diagram. You can see the vesicles. So the vesicles, it carries the proteins, lipids which are being modified and then finally packed to be carried to the final destination. Okay. So packaging is done here, right? And this is followed by the third important process called as So we were talking about the Golgi apparatus function, modification of proteins, packaging of proteins, lipids into vesicles, and then secretion. Secretion of proteins, this is the function in which it is responsible for sending the protein to their final destination. Okay, responsible to send to final destination. Next function is formation of lysosomes. Formation of lysosomes. Okay, so all of us know lysosomes, these are the small vesicle kind of a structure, and Golgi apparatus is the one which is responsible to form this ribosome. So one of the NEET update or else NEET fact is that if you are asked which is the factory of lysosome, okay, factory of lysosomes, your answer should be Golgi apparatus. Okay, Golgi apparatus or Golgi body or Golgi complex, whatever. Okay, be very clear with that. Golgi body is called the factory of lysosome. Okay, another function is cell wall material formation. Cell wall material formation. Okay, now obviously because it is a cell wall, this you can see only in plants. Okay, so this is about the functions of Golgi apparatus: modification, packaging, secretion, formation of lysosome, and then cell wall material formation. So when you see the flow chart of this, the material or else the information starts from nucleus. Okay, it is being given to ribosomes in the form of mRNA, messenger RNA. It reaches to ribosome, and this ribosome goes and gets itself attached to the rough endoplasmic reticulum. Okay, the rough endoplasmic reticulum transcribed your proteins or your information into proteins, and then from the rough endoplasmic reticulum the material is transported to Golgi body. In the Golgi body it is being modified, packed, and then it is given into vesicles, and this vesicle carry the particular protein, lipids into the final destination. So this is how the cell transport system works. Hope the class was clear, students. See you in the next class. Do subscribe, share and follow our classes for more such videos. Thank you for watching.
Hello students, so we have seen this in detail, that Golgi apparatus, the structure and function. So its main function is to receive the product, that is the protein and lipids, from the endoplasmic reticulum, pack it, okay, and then label it, and then give it to the respective destinations, okay, in the form of vesicles. So this endoplasmic reticulum has two ends, the cis facing end and the trans. Cis is the receiving end, which is also called as oral, which receives the product through the incoming vesicles, and then trans is the shipping or as exit gate from where the vesicle packs the product into itself and then gives it to the different location of the cell. So this is how the Golgi apparatus work. So hope you like the class. Do share, subscribe and like the video so that I can bring more and more videos like this for you. Thank you.